There we go. Yay. She can be taught. Okay. So I'd like to thank everybody for coming, and the New York Microscopical Board of Managers is really happy to bring you a seminar today. We had to schedule it midday because one of our speakers, Emily, is in South Africa, but we are recording it, and we heard that recording start. And we're going to be putting it on the website for members who can't make it today. Speaking of the website, thanks to our current president, Deb Capel, and her team, the website is back up and running and has our lecture listed on the events page. And there are a few other things, so make sure you check it out. The societies had some difficult years with COVID. Certainly, a lot of societies had issues during that period. And the sprinkler system at the headquarters flooded during COVID period. So it's been a rough couple of years, but we're trying to revitalize the society. And so please think about folks that you'd like to invite to join the group. If you have ideas about potential events or speakers, please send them along. We'd love to hear about them. So then, without further ado, let's begin the seminar. We've designed the seminar to be about 40-45 minutes. And I suggest you hold your questions till the end unless there's something that's very unclear or there's some kind of a technical glitch. I won't be monitoring the chat because I have to nix multitasking at this point in my life. But we will save it. You've received the short speaker bios, but rather than reading them to you, I'd like our speakers each to take a minute to give a wee bit of their background so you can connect the face with the presenter. So I'm Renate Reimschuessel, and I graduated from University of Pennsylvania vet school. I did a lot of rotations in exotic animal medicine. Then I went into private practice and did dogs and cats and whatever came in and whatever weird came in, I would do. And then after about 5 years in practice, we moved to the Baltimore area, and I looked around and I got a PhD in pathology, focusing on fish. And let me just move that. There we go. Then I stayed at the University of Maryland and worked in aquatic pathology. And then FDA, Food and Drug Administration, offered me a job setting up their aquaculture research program. And then later, and I'll go more into this during the talk, I set up a network of labs to work with Food and Drug Administration. So that's my nutshell. Emily?
Well, I'm an American, but my family moved to Africa when I was 4. And apart from going to college in America, I've lived in Southern Africa my whole life, more or less. I trained as a vet in Zimbabwe and a pathologist in Zimbabwe under Nancy Koch, a pathologist who had come out from UC Davis. And I've spent most of my career as a wildlife pathologist, working either for myself or at the zoo, and now I'm currently at the University of Pretoria at the Faculty of Veterinary Science. Can't hear you.
And Sarah?
Hi, my name is Sarah Nemser. So I graduated from University of Maryland, and I have a master's degree in food science. When I was in graduate school, I worked at the FDA. I was an intern there at the Center for Food Safety and Applied Nutrition, so CFSAN. And that's where I learned a variety of analytical chemistry techniques, and I learned Raman spectroscopy. And then when I finished college, I got a job at Center for Veterinary Medicine working for Renate, and I am the network coordinator for the VetLearn program, which Renate is going to tell you all about. And I've been there for the past 10 years. Thanks.
Great. Okay, so let me share my screen and start the PowerPoint. Let's see. There we go. Where is it? Let me see here. And okay. So one thing that, because Sarah is working at Food and Drug Administration, it's important that we mention that the views expressed in the presentation are those of the authors, do not necessarily reflect the official policy of Department of Health and Human Services, U.S. Food and Drug Administration, or the U.S. government. So, okay. So let me move the people. How do I get rid of? Let's see. There we go. All right. How did this all come to be? In 2007, Center of Veterinary Medicine at the United States Food and Drug Administration had the largest pet food recall in history. Thousands of animals were impacted. There were many calls to FDA, over 11,000 calls in a 2-week period. And usually there were about 5,000 calls a year up until that point. So it was massive inundation. And eventually the culprit was found to be melamine and cyanuric acid, and it was present in wheat gluten that was imported and used to make pet food. It was an adulteration, and the melamine by itself is relatively non-toxic, but in the body, melamine and cyanuric acid combine in the kidney and form crystals. And those crystals plugged the microscopic tubules in the kidney and caused kidney failure. And you can see in the lower right, those are the kind of crystals, and you can see the outline of the tubule there, those cells around it. And it basically plugged the plumbing and caused kidney failure in quite a lot of pets. So while I was working with this, I visited FDA's Forensics Center in Cincinnati, Ohio. I observed them using Raman microscopy to identify the tissues, the crystals in the tissue sections. And I brought along samples of pet cases. And at that point, we had also dosed fish and pigs with the melamine and the cyanuric acid. We needed both compounds to create the crystals experimentally. And the Forensics Center identified the composition of the crystals as melamine cyanurate. So during that time, we collaborated with a lot of pathology departments around the country to get specimens. We worked with UC Davis, with University of Florida, with Iowa. There were pathologists there that were quite willing to try to help FDA figure out what was going on, because this was a really new thing, and we needed as much expertise as possible to help figure out the problem. So after this event, CVM saw the need to work with other veterinary diagnostic labs.
Renata, did your slides jump to the next one? Because I'm still just seeing the main slide, and I want to make sure everybody else.
Oh,
see
it
the
should
slides.
be progressing.
Okay. I might
It's
leave and come
progressing
back.
for us.
Okay, it must be just me. I just wanted to double check. Thanks. Sorry.
I can stop share and try sharing again. Let me do that for right now. Let me try just to make sure everybody's, let's see. Oh, that's right. Oh, screen sharing stop. Okay, I screwed up again. Let's try it. Oh, I closed the whole thing, that's why. Are you with me?
Yeah.
Should be loaded. Do I hear a yes? Oh, it's good. All right. Okay. So in 2010, we actually got funding to develop a network of laboratories to work with the Center for Vet Medicine. And up until then, I had been, in charge of aquaculture, and I switched doing aquaculture research to working more with the melamine work that I had started in 2007, basically dropped everything during that really serious event. And in 2010, when we got the funds, I started developing the network, hiring staff, and that's how I got Sarah to join us, and getting equipment, and finishing up the research on melamine that I had started during that pet food event. And in order to work with tissues and crystals, I said, well, I'd really like to start Raman technology at CVM. So I did start a network called VetLearn, and I'll tell you a little bit about that. VetLearn's mission was to advance CVM's mission of protecting human and animal health by coordinating a network of diagnostic veterinary labs. There is a consumer complaint system at FDA, and we responded to various complaints that people would have. We'd respond to adverse events believed to be related to animal food, look at product problems involving animal foods, and also receive reports for large animals. So it wasn't just pets, but also for livestock and horse feed or exotics. The general scheme of things is when you have an animal that's sick and you suspect that it's a food or a drug, you can submit a complaint to FDA, and now they have an online portal. And there are three basic actions. They can initiate regulatory action, they can monitor for similar complaints, or they pull in the network and have VetLearn gather more information. And that's done through a variety of ways. We read through the complaints, we talk to the vets, we talk to owners, and then after getting more information about what happened, we then may actually, or VetLearn may test animal samples at network labs. And you can see here, there's the United States, and there's quite a number of them. And most of these are either at Department of Health labs or state or vet school labs. And the focus there is more on the animal products, tissues, blood, fecal samples, whatever, rather than on food testing. And the basic question is, was this caused by the food? And do we have evidence to that effect? So doing this, setting up the whole network, involved talking with a lot of people and at various schools. And Molly Church was a pathology resident at UC Davis, and she requested VetLearn to look at some of these renal crystals. We had the equipment, we had personnel who knew how to use it. We had seen cases of ethylene glycol in pets where the cases had developed crystals. And Molly said, I've got a fair amount of cheetah samples with renal crystals, and can you guys actually clearly identify them? They were presumably ethylene glycol, but they're, the UC Davis didn't have a Raman to actually investigate those. And So one of the reasons that I said this is a good thing for VetLearn to do was that you can have food affect renal crystal formation. And also it's important for the lab itself to maintain their expertise by looking at samples, and you know, we would get cases far and few between, and this was a good way to hone our skills again. And so we said, sure. So the collaboration began. And this is just a diagram of FDA, and you can see there's the Center for Veterinary Medicine where VetLearn is located as part of that. There's also CIFSAN, and that's Center for Food Safety and Applied Nutrition. And we had a ramen at CBM, and we began the collaboration. But as you know, with many studies, scientific studies, you start, you make some progress, and then sometimes they're on hold for a little while, and then you get some more information, and then you can pull in additional people based on their expertise. And at Center of Food Safety and Applied Nutrition was where Sarah Nemser had actually worked when she was an intern, and Dr. Betsy Yakes had a system there, a ramen system, and she had, you know, a lot of expertise and had been one of Sarah's mentors. So we pulled her in as well and said, you know, can you help us get really good scans of these? And so that's how we continued with an additional group at FDA. And then Molly Church was moving to different places in her career. And we ended up being in touch with Emily Mitchell, who then inherited a lot of this investigation with us and began working with the group that had initially agreed at FDA to do that. So I will turn it over to you, Emily, and I'll move the slides. Just tell me when.
Okay. Okay, so the late Linda Munson was a wildlife pathologist at UC Davis and University of Tennessee before that. And she put together a network of wildlife pathologists or wildlife veterinary pathologists who were working on cheetahs, which was one of her key species that she worked with. So there was a network with I actually attended a conference, a veterinary pathology conference, and I spoke to her about it. And we agreed that we would all get together and try and figure out what we could about the cases that we had. And the people who collaborated were Karen Theriault, who's at the University of Illinois, Karen Lemberger, who is at a private diagnostic laboratory in France, and myself. And we had, I mean, every now and again we saw cases and we didn't really know what caused them. We still don't know what caused them. But there was a dramatic increase around 2004. And because cheetahs are an endangered species, we really needed to figure out, if we could, what was going on. But we needed the numbers of animals. We needed to combine our efforts to try and figure out the epidemiology and have decent numbers of animals. Thank you. So we started out by trying to figure out why they had oxalates in their kidneys. Some, you know, in kidney disease, you do get some oxalates, but these were really dramatic numbers and significant renal tubular damage. And one of the possibilities was that somehow the cheetahs were taking in more oxalates or the precursors of oxalates. And of the list that's there, nothing really made sense. We even investigated propylene glycol, which was used is used in animal food, but we found no evidence of it at all. The only one that is possible is if you feed animals a collagen-rich diet, so a lot of skin, a lot of connective tissue in their diet, that would increase the amount of oxalates in the blood. we thought we looked at the possibility that they were, for some reason, increasing the absorption of oxalates from the intestine. They mostly don't have cheetahs and between the three of us, we had lots of experience, but they mostly don't have intestinal disease, and obviously we don't do resections very often. But one of the possibilities is that they do get a Helicobacter-related, if not caused, gastritis, and they get treated with antibiotics. And we read some literature that said if you the antibiotics reduce the number of oxalobacter species in the intestine and they break down oxalates, it was possible that you would have a hyperoxalemia and other dietary factors. Again, related to collagen, if there are genetic mutations that reduce the efficacy of digesting collagen, and that's why some domestic cats and miniature schnauzers and people have oxalate nephrosis. And then there were some other things like vitamin deficiencies and liver disease, which we don't really see. And then if you have decreased urine volume and changes in urine pH and calcium content, that can increase crystal formation. But there was no reason to think that those were a feature. So we were a bit puzzled, and the first thing we wanted to do, especially in view of the melamine issue, was confirm that it was oxalate, which we thought we could see. So next slide. So I had a database of 100 cheetahs. I've been doing cheetah postmortems since 1998. There were 50. We had, those are the controls actually. So we had in my database, I had 56 animals with significant numbers of, with any oxalates, and 110 that didn't. Caranterios was 40 and 50, and in France, actually that's a typo, that's my mistake, it was 4 and 4, or 4, we didn't have a control from her side. And so we sent samples from all 3 locations, all of the French samples, and a selection of samples from America and Africa, to have Raman spectroscopy done. And our results were, I think there's a click there somewhere, there should be, yes. Our results were that basically virtually all the crystals that were seen were oxalate crystals. I think that's the end of my section.
Okay, thank you. Oh, and I just wanted to, for those who are not as familiar with kidney, just point out these are the tubules, and here are crystals within the tubules. These are red blood cells, and you can see the renal tubules here without the crystals. Okay, Sarah.
Thanks. So we'll kind of take a step back here now to just talk more about Raman before we dive into what we're going to see with the results and more crystals and pictures and things that I hope will be interesting to folks. But here's a picture of our instrument at CVM. This is the Raman Hariba Explorer instrument, and we did purchase this back in 2009 in response to the melamine pet food recall that Renate had talked about. So we'd use this instrument to identify melamine and cyanuric acid crystals in the animal samples that were directly related to that recall. And then we'd also used it in a number of different studies related specifically to melamine and cyanuric acid with the renal crystal formation. So we were familiar with looking at crystals. And then, as Renate had talked about, we had several consumer complaint follow-ups, and one of those cases did involve suspected ethylene glycol poisoning, and that was in a dog. And so we had the crystal from that case, and we used that to analyze it on the Raman, and that gave us our control case for this work. So we had crystal that we knew was calcium oxalate crystal, and I'll get into that as we get into kind of what the pictures and everything looks like in the results. But something to note that might be of interest to folks is that this specific Raman is using confocal Raman microscope. And so that allows us to have a full 3D spatial resolution, and we can analyze discrete volumes in a transparent sample. This property is really useful if you're looking at layered samples, inclusions, or thin samples on a substrate, such as cells or tissue on a microscope slide. And that's what we were looking at. You can also look at materials in glass and plastic containers, like liquids in a bottle. So this Raman setup is suitable for in situ, in vitro, and in vivo analysis. And also the Raman is a non-contact, non-destructive technique. So you can look at the crystals and not destroy them. We did burn some of the other material around the crystals, but we were able to get the crystals and look at them. And also the tolerance for the Raman for water was really important for this analysis. So it's a great tool that we can use. And this is our instrument. And then next slide, this is the instrument over at CIFSAN, as Renata had mentioned, that Dr. Yakes has. So this is the Nicolette Almega XR, which is also equipped with that confocal microscope. But Dr. Yakes' Raman is definitely more advanced than our instrument. The Explora is kind of, like the base level, and then this one is up there, and it has a lot more features with the library and analysis portion for this instrument. So we use this instrument to do the final testing for the work. So next slide. All right, we're going to jump into some Raman spectroscopy fundamentals. Some of you might be familiar with this, and you probably could explain it better than I can, but some people, it might be new to you, so we'll kind of go through the basics and point out how things are working. So Raman is a vibrational spectroscopy, and when you're using vibrational spectroscopy, analyze the samples chemically. You can use light to create or excite the molecules and make things vibrate, and you can interpret that interaction afterwards. So for example, if you have a green laser pointer, you point the green laser pointer at the wall, you see the color green, and that's the scattered light. It's coming out as the same color. So this phenomenon is called Rayleigh scattering. Now there's also the Raman effect, and that is inelastic scattering that leads to emission of light at a different wavelength. So this is vibration on the molecular level. Now it was predicted in 1923 by Adolf Smeckel, but it wasn't discovered until 1930 with C.V. Raman, hence the name. And Raman couldn't really go anywhere until the laser was developed. So it couldn't move forward until the laser was discovered. And at that point, then samples are irradiated with laser and the light is scattered and analyzed with a spectrograph. And this information is what results in the Raman spectrum and the characteristic signals or bands that is what we're looking at for the material. So the next slide. This slide here can show the difference between Raman and Fourier transformed infrared, so FTIR. FTIR is the gold standard for molecular spectroscopy, but Raman offers some unique and several advantages over IR that were critical to what we're doing. So in this picture, you can see we have two different compounds, and the black peaks in the graphs are the IR, and then the blue is what the Raman looks like. So we're looking at those peaks to tell us what we're seeing and what the compounds are. The different structure will come up with different peaks, and that's how we can identify things. So Raman has several advantages, and it avoids interfering compounds such as solvents, cells, and it's easier when it comes to sample preparation. And then it also offers the ability for depolarization studies, which was really useful when we were looking at these samples here. Next slide. All right, I'll get into sample preparation. Now, we had the archived formalin-fixed paraffin-embedded kidney sections, thanks to the different groups that Emily mentioned. And we were ready to prepare our samples from these, but you might guess that paraffin and dyes might show up on the Raman, and you'd be correct. So we needed a way to have the samples where we didn't have the paraffin interfering or any dyes that would cause extra peaks that were not what we were looking for. So we used unstained deparaffinized sections, 6 to 8 micrometers, I think, on quartz slides. And we used quartz because we didn't want to use glass, because glass will also produce a peak. So we use quartz. That's great for ramen work. When we prepared the samples, we asked the lab to make 1 H&E slide and then 2 unstained. So we used a lot of slide mapping where we looked at the H&E slides to find the crystals, and then we used the actual unstained slides on the Raman instrument so that we wouldn't have that extra interference. So the next slide. Okay, so this slide is zoomed out really far, but you can see it's pointing to the very, very tiny and bright crystal here. This is a polarized picture of what we were looking at, and this is one of the H&Es. And then if you go to the next slide, we're zooming in a little bit more and pointing to the crystal as you can see it. And then the next slide, we've zoomed in even farther. So we were able to find the crystals in the material, and we would use the H&E to find them. And then I'll show you in a little bit what it looks like on the Raman. The Raman microscope is not quite as easy to use, and so we needed to have the H&Es and the slide mapping to help us point to where we were going to go as we were moving around with the Raman instrument. And if you go to the next slide, here we have this crystal polarized. Okay, next slide, I'm going to talk just a little bit about the control case that we used. So we did have a case of confirmed canine ethylene glycol toxicity, and we used that as the control. And we were expecting to see calcium oxalate crystals. So that's what the control was, and this was from a previous case. And the next slide shows us that known positive. So here's the H&E from that. And then if we go to the next slide, you can see the crystals polarized. So here we have a lot of crystals that we can take a look at. And these are what we looked at on the Raman to get our spectra for the control case. The next slide has what it looks like on the Raman. So this is the Raman picture that shows up on the computer. As you can see, it's really dark. It's kind of hard to see the structures. So we had to use big landmarks to try and get to where we were going to actually find the crystal. And then we've zoomed in on the right-hand side, and you can see what the crystal looks like on the Raman through the microscope, looking at it on the computer. So it's very bright. And once you find it, you know that you've found it, but it's quite difficult to get to where you're going. The next slide shows us the known positive on the Raman. So we have the peaks of interest here, and we're comparing this to our calcium oxalate control, and that is the blue peaks, the blue spectra here. The red is the control sample, so the known calcium oxalate crystal. And interestingly enough, and we'll see this as we look a little bit farther, when we're looking at the peaks that are around the 1400s, so the 1489 and the 1462, you can see for the monohydrate, the two blue peaks are kind of the same. And then in the control sample, we've got just one really large peak at the 1489. And we saw when we were looking at all of these crystals that the peaks would kind of flip-flop back and forth, and maybe the right side would be stronger or the left side would be stronger, or sometimes it would just be one single peak. So you'll notice that when you're looking at the final results and what we came to. And it really just depended on the orientation of the crystal, it seemed, how those peaks were coming out. So this is what it looks like. This is what we were hoping to find when we looked at the cheetah crystals. And we did see this, but this is the control compared to actual calcium oxalate control monohydrate with the powder. So powder versus in tissue. If we go to the next slide, here we have the actual sample. So this is what it looks like with the cheetah. You can see I'm pointing to the crystals here, and they look quite large in this. And these ones were a great representation of what it looks like. Sometimes they were much more difficult to find. And if you go to the next slide, we're zoomed in here and you can see what they look like in the sample. If we go to the next slide, we have results and comparing everything together. So you'll see the blue spectra, that is the calcium oxalate monohydrate powder. And then the red is that control sample with the known ethylene glycol poisoning calcium oxalate crystal. And then the purple is the cheetah sample. So this is what came up when we looked at large majority of the cheetah samples. So we can see that there's a correlation with calcium oxalate. If we go to the next slide, the next two slides I'm going to show you are just really nice examples of pictures and what we're looking at when we're viewing things on the Raman. This is very polarized, so we can really see and find the crystals, which made it a little bit easier. They didn't all look like this. This is just a really nice example with the crystals present. And then if you go to the next slide, it's zoomed in on a great representation here of one of the crystals. On the next slide, we have the results. So this is what was actually published in the paper, where we are providing information on what we're seeing with Raman. And we have all of the information in the different bands and the different carbon and oxygen that we're seeing, what's causing the peaks in the crystal, and what we can see. And so I won't read you all of the different peaks, but you can take a look at this afterwards and you can see how everything matches up and lines up. And so we saw this in numerous samples where we're showing this representation of the strong correlation to the calcium oxalate. And the next slide is the conclusion. And I think at this point, I'll turn it back over to Renate and she can add anything else. Thanks.
Cool. Thank you. So, basically, Raman is a tool to help gather more information about this issue. What you saw with Sarah, sometimes you have, a whole buco load of crystals and you can find them easily. Other times you really, you're looking at each glomerulus and saying, well, this is a serial section and the crystal should be in this one location on this slide. So Raman, many times when people look at drugs, they see, they have the powder on the slide, and it doesn't matter where you look, you can find it. So when Sarah did that, I mean, she was meticulous, and it was sometimes like an Easter egg hunt that was really looking in the long grass. So it doesn't give us necessarily the answer to why are there oxalate crystals, but by looking at so many of the kidneys, we certainly confirmed that, for the most part, those crystals that were being observed for many years by the pathologists in the field were calcium oxalate and not a different time. For example, one of the rule outs would have been melamine cyanurate, but there are other kinds of crystals as well in kidneys. So we are now quite confident that what the pathologists have been seeing throughout the years have been the oxalate crystals. Unfortunately, the jury is still out is exactly what is causing that. And Emily and her group are still working at it, and they've published quite a number of articles on, various potential aspects that could increase the likelihood of developing the oxalate crystals. This paper is online. I think it's open access, usually, yeah. So you can certainly download those. And I guess at this point, I'd like to open it up for questions. And I'll look in the chat, just make sure that nothing's sitting in there. Anyone have questions or comments?
Okay, about ramen. So lasers are by nature polarized. Is the stage rotatable so that you could optimize the contrast of those crystals?
Oh, you're muted.
Sarah?
Yeah, thank you. So the ramen that we have on the bottom, it has a disc, yeah, that you can turn. And so then you can line everything up properly and have the polarized view. So it's a really useful tool to have that feature there. Yeah. Looks like there's another question.
Yep.
Yeah.
Go for it.
Right. So, are other animals sensitive to oxalate in the kidneys, like the cheetah?
Yes, they are. Domestic cats, we've seen increasing numbers of domestic cats. And I've seen it in antelope, I've seen it in a wide range of species. It's just that it seemed to be, we seem to suddenly have an increase in cheetahs in South Africa. And because they are endangered, we had significant numbers to examine. That's why we focused on the cheetahs.
What about humans? Are humans also sensitive to?
Yeah, they are. Obviously, accidental ethylene glycol or malicious ethylene glycol toxicity will do that to people, but also the genetic defects in carbohydrate and in collagen metabolism can result in it. And it's quite interesting because it's quite episodic. You know, you may have a predisposition and then something will happen and trigger an episode of kidney damage.
Since the cheetahs are endangered and their population is small, is that you think it's partially due to a lack of genetic diversity?
We don't really think so, because one of the few things, because whatever we looked at, we couldn't really find any particular cause. But one thing that we did find was that there was quite dramatic geographical clustering. And that makes it unlikely, because all the cheetahs everywhere in the world are relatively closely related. You can do skin grafts between cheetahs, for example. Because they're all relatively closely related, we didn't think it made sense that it was because of inbreeding, because the levels of inbreeding are not particularly higher in one institution than in the other. But some institutions have a lot of cases, some have sporadic, and some have had none.
There's a comment in the chat that their orange tabby just had sodium oxalate crystals in his kidneys.
Let me just unmute. We think they're in about 2000 and in the early 2000s, the formulation of cat foods changed because cats on dry foods anyway were particularly susceptible to a different kind of crystal, and they changed the formulations of the domestic cat foods to address that. And we are not sure whether we started seeing the changes in the domestic cat population because of some way in which the domestic cat population, the domestic cat food was being formulated. We still don't know if that's true.
Yeah, cats are notorious makers of this very sandy, gritty material in their urine that, commonly struvite crystals. And, the males have just such a narrow opening that it's it's a common emergency for veterinarians to have to deal with urinary obstruction in cats. So, but the oxalate crystals are a different story. Yeah. Great. Any other questions? Well, I'd like to thank New York Microscopical Society for the opportunity to share this information, and I'd like to thank very much Emily and Sarah for their work on this issue and for being willing to take busy time of their days and Emily at night to join us and to give this presentation. So a reminder to the folks that are online, and then those who might be watching this on the recording, please, send us your ideas for lectures. We'd like to keep the society active, and I think this was a successful venture today, and I appreciate everyone coming online and listening. So I'm seeing some notes in the chat, and again, I can send out a link to the paper too if folks are interested in that. All righty. So I guess, Julie, you can end the meeting for everyone. Thank you so much.
Hi, thank you.
Bye-bye.
Thank you.